So it is a pleasure for us to have today with us in our Bar-Ilan University Vision Science Seminar, Dr. Zvi Roth from the Gonda Multidisciplinary Brain Research Center at Bar-Ilan University. Dr. Roth did his BSc in Physics and Computer Science at the Hebrew University, where he already took part in the research of Professor Chaim Sompolinsky's lab in the Rakach Institute of Physics and ELSC, which is the Edmond Elin Safra Brain Research Center at Hebrew University. He then did a PhD in brain sciences on computation and information processing at ELSEC under the supervision of Professor Udi Zohari in the Hebrew University, where his thesis was about brain representations of visual percepts. He also did a summer course, on computational neuroscience and vision at Cold Spring Harbor. After this, he did a postdoc at the Lab of Brain and Cognition headed by Dr. Elisha Miriam at the NIMH in NIH. And then he continued there as a research fellow and also furthered his research as a research fellow in the School of Psychological Sciences at Tel Aviv University, working also with Professor Liad Mudrik. Recently, he opened his own lab named Lab of Brain Representations at the Gonda Brain Research Center at Bar-Ilan University. He won multiple awards as the ELSEC Prize for Outstanding Academic for Published Paper and the NIH Fellows Award for Research Excellence. He publishes in leading journals as Nature Communications, eNeuro, eLife, JNeuro, and more, and he also wrote a book chapter. In his research, he studies how the brain represents the visual world, focusing on how many different factors, both internal and external, affect neural representations in the visual cortex. His aim is to reveal how these lead to and underlie visual perception and shape perceptual representations. He uses non-invasive measurements of human brain activity, mainly functional MRI data, with perception and where the methods and principles he uncovers can be generalized to be applied across the brain. So without further ado, I'm so happy to welcome you, Zvi, to our seminar to hear about your exciting research, and thanks so much for joining us.
Thanks a lot, Shalom. So in my research, I use fMRI experiments, as you mentioned, combined with computational modeling and other methods to study the human visual cortex. How visual features are captured by neural tuning, how neural activity reflects and represents visual stimuli. And I'm particularly interested in cases where the tuning or the representations change over time. And this can happen as a result of learning. It might be the case that changes in the internal state, such as stress or arousal, such changes might alter sensory representations. But today I want to talk about changes that seem to be detached from any obvious learning or behavioral changes, continuous changes over long periods of time. And this phenomenon is called representational drift. And the work I'm going to present is from my postdoc with Eli Miriam at the NIMH. So I'm going to briefly describe what I mean by representational drift, and I'll then show you evidence for drift in human visual cortex. We'll then discuss how drift relates to repetition suppression, and I plan to leave time for questions, but feel free to stop me during the talk as well. So representational drift has been observed primarily in mice, and it's been reported in many different brain areas covering very different types of representations, including the hippocampus, posterior parietal cortex, olfactory cortex, and recently also in mouse visual cortex. And the common finding in all these studies is that representations continuously change over relatively long periods of time, so hours or days or even weeks. Now, first I want to point out a difference between drift and noise, okay? Because of course, when we measure neural responses repeatedly over time, the measurements will vary. They won't be identical because of noise, right? But that doesn't necessarily mean that representations are changing. So let's see how drift differs from noise. If we just have noise, then we'll see a difference between responses at time 0 and at later times, at later measurements. So on the left, these black lines show some neural activity measurements taken at time t0, t1, t2, and they're normalized to time t0, so we're only seeing the changes, right? And here the measurements fluctuate randomly. Now, as a result, on the right, you see that if we compute the similarity between responses measured at time 0 and responses measured later on, the similarity drops. The measured responses will not be identical. Okay, that's noise. Now, representational drift, on the other hand, means that responses are changing systematically and continuously over time. Okay, so they're not only random fluctuations, but there's also a systematic drift. So some neurons, in this case, have increasing responses, others have decreasing responses. And as a population, the responses are becoming less and less similar to the original responses measured at time 0, as you see on the right. So that's the idea of representational drift. And as I mentioned, it's been demonstrated in several regions of the mouse brain. And in 2021, two studies came out showing drift in mouse visual cortex. One of them was from Yaniv Ziv's group at the Weizmann Institute. And in this study, they analyzed data consisting of both calcium imaging and neuropixel recordings from mice passively viewing natural movies. Now, natural in the sense that they're actually filmed, okay, they're not gabors or gratings. And this 30-second movie is repeated many, many times within each recording session. Now, in order to measure representational drift, they computed correlations between population responses. So they take the population response vector, okay, the vector of all the neurons' activity at each time bin of the video, and they calculate the correlation with the population response to the same time bin in every repetition of the movie. And they do this separately for every time bin. And finally, they average the correlation across all time bins. So that gives them a correlation matrix between all pairs of movie repeats. And you can already see here that the correlations are higher around the diagonal. Okay, more orange, more yellow around the diagonal. So that means that when the pairs of repeats are closer in time to each other, correlations are higher. They then looked at correlation as a function of time between the repeats, and you can see that as the amount of time between repeats increases, the correlation drops. So this means that representations in mouse visual cortex are changing or drifting. Okay, but mice have a visual system that's quite different from humans, right? We rely on our sense of vision, on sight, much more than mice do. So does human visual cortex exhibit representational drift? Now, it's important to keep in mind that we're gonna be answering this question with fMRI, and we know that voxels are measures of population activity. It's possible that there is drift at the neuron level and not at the voxel level. And also, as already mentioned, there are very likely differences between mice and humans. But so to answer this question, we turned to the natural scenes dataset, which consists of a large amount of 7 Tesla fMRI data, measuring responses to natural scenes in 8 subjects, each of them scanned between 30 and 40 sessions, viewing up to 10,000 different scenes over many months. So each image out of the 10,000 images was presented a total of 3 times over the entire experiment, okay, 3 times over all the sessions. So in each session, a very different set of images was presented. And the task was a recognition memory task. Subjects pressed one button if the image was new, if they hadn't seen it before in this experiment, and a second button if the image was not new, if they had seen it before in the experiment, either in the current session or in a previous one. And there was also a separate session for retinotopy mapping. So how do we use this data set to test for representational drift? So we used a model fitting approach. And this is a model that was designed for primary visual cortex, for V1. So we're going to be analyzing V1. Okay, we took all the 10,000 images and we first passed them through a steerable pyramid model, which consists of a set of filters where each filter corresponds to a certain spatial frequency and orientation. And the filter response tells us how much energy each image has for each combination of frequency and orientation. Okay, then for every voxel in V1, we take the voxel's receptive field, the PRF, which was estimated in a separate session, and we use it to sample each filter's response. So this model output tells us how much energy at each orientation and spatial frequency falls within the voxel's PRF. Now, we also have the voxel's response amplitude to each image. So we perform multiple linear regression using the outputs of each model filter to predict the voxel's responses. And that gives us, for a single voxel, weights for each model filter. Okay, basically that tells us how much each spatial frequency and orientation determines this voxel's response. Okay, it's a bit like a two-dimensional tuning curve for orientation and spatial frequency. Now, once we have the weights for one session, say for session number 1, we cross-validate. We use the weights to predict responses in a different session, say session 2, which, remember, it includes an entirely different set of images. And we then compare the predicted responses to the actual responses. And this way, we get cross-validated R-squared values for each voxel, which quantify how well the model generalizes from one session to another. Now, what I described here is for a single pair of sessions, sessions 1 and 2, training on one and testing on the other. The next step is to train the model on each of the 30 sessions and test it on all the other sessions. And that gives us a matrix of cross-validated R-squared values, goodness of fit values, for training the model on every session and testing it on all other sessions. And if we just look at cross-validated R-squared, or I'll call it CV R-squared, as a function of the number of sessions between training and testing, which we'll call delta session, then if there's no drift, the representations are stable, then CVR squared should not depend at all on delta session. Okay, we should just see a straight line just fluctuating with some noise. But if increasing the number of sessions between training and testing causes a decrease in the model goodness of fit, then that means that representations are changing over time. And it turns out that there is drift. Okay, CVR squared values decrease with delta session. So here in this third panel, each colored line is a single subject, and the black line is the average. So the model predictive power drops with time. Now, to test whether this drift is significant, we used a permutation test, shuffling the order of sessions. And here I'm showing you the null distribution in gray, and the actual amount of drift is the black bar. And you can see that there is a negative correlation, meaning that CVR squared decreases with delta session, right? Those lines are slightly dropping. And the point here is that it's more negative than the entire null distribution, meaning that we wouldn't expect to see such an effect by chance. Okay, so this drift is significant. I want to stress that for this analysis, we're not relying on repeating a stimulus over and over again. Okay, instead, what we're trying to do is to capture the voxel's response through a model, similar to a tuning function, and we can then predict the voxel's response to any stimulus. And we can also tell whether the tuning function is changing over time. And the decrease that we see here isn't a decrease in the response amplitude, it's a decrease in the model generalizability. Okay, now, that decrease in generalizability means that something about the tuning in V1 is changing over time. So we want to know what aspect of tuning is changing. Now, if you imagine a voxel's tuning curve to some stimulus parameter, say orientation, it might look like this, some sort of bell-shaped curve. And our finding of representational drift implies that this curve is changing in some way over time. So what would that change look like? Well, there are four possibilities. So first, the response baseline could additively shift up or down. Okay, so say the voxel initially has the tuning curve shown in blue, and over time the entire curve is shifted down. Okay, that's one possible change. Alternatively, the gain could multiplicatively change. Okay, so become larger or smaller. Or the tuning width could change. It could become broader or narrower. Or the stimulus preference could change. Okay, it could shift from one preferred stimulus to preferring a different stimulus. Now, how can we distinguish between these different scenarios? How can we determine which is the correct one? Well, let's note that the cross-validated R-squared measure is sensitive to each and every one of these changes. Okay, a baseline difference, for example, between predicted and measured data, if there's such a difference, it will result in a lower R-squared, as will a gain change, or width change, or a preference change, right? And the larger the change, the smaller the cross-validated R-squared. On the other hand, Pearson's correlation is sensitive only to two of these changes, right? To changes in width and preference, but not to changes in baseline or gain. If there's only a baseline difference between two time series, or only a gain difference, one is a scaled version of the other, then they'll still be perfectly correlated. They'll have a correlation of one. And notice that until now, we quantified the goodness of fit between the predicted and measured responses with CVR squared. But if we instead measure the model goodness of fit with Pearson correlation between the predicted responses and the measured responses, we find that the drift disappears. So you can see here in the middle panel that the goodness of fit doesn't decrease with delta session. And on the right, the empirical measure is right in the middle of the null distribution. So there's no drift here. And this is because R squared and correlation are different measures of goodness of fit, right? Correlation ignores changes in the mean or the variance, while R squared is sensitive to those changes. So if there's a difference in the mean or the variance between the prediction and the measured response, CVR squared will be low. Okay, so this means that the drift is due to changes in either the response amplitude mean, which is equivalent to a change in the tuning baseline, or due to changes in the response amplitude variance, indicating a change in the tuning gain. So which is it? Is it changes in the mean or changes in the variance, or maybe both? So to tackle this question, we normalize the sessions either in terms of the mean response or the variance. Okay, because a baseline change will affect the mean response, while a gain change will impact the variance. So first we normalize the variance, scaling the response amplitudes, the fMRI betas, so that every voxel in every session has a variance of 1. And it turns out that normalizing the variance has no effect on the drift. So when all sessions have the same variance, there still is drift. Okay, so this indicates that a change in gain is not the cause of drift. And next, we normalize the mean by subtracting the mean response amplitude from every session for every voxel, so that now each voxel's mean response amplitude is 0. And it turns out that normalizing the mean completely removes the drift. And this indicates that each voxel's mean response is changing across sessions, and it's that change that underlies the drift. So these results indicate that the drift is due to the baseline of voxels tuning curves changing over time. So single voxels are changing the baseline of their response to visual stimuli. Okay, but what does that mean for how V1 represents a stimulus? Visual perception likely relies on the response of the entire V1 region or even the entire visual cortex. Does the representation across the entire population of V1 drift as well, or does it remain stable? So to test this, we take the model fits for all V1 voxels from one session, and we simulate how the model expects them to respond to a single image. Okay, so we get an estimate of how V1 represents an image in a single session. Then we do this for every session, and we then compute the correlations between the population responses for each pair of sessions, which gives us a correlation matrix for a single image, here, image number 1. Okay, we do this, this is for image number 1, but then we repeat that for a bunch of images. And finally, we take the average of all those correlation matrices. So each element in this matrix is a measure of how similar population responses are between two sessions. And the question is whether, as we increase the number of intervening sessions, the delta session, right, does the correlation between these representations drop, or does it stay at roughly the same level? And it turns out that population responses drift as well. They're changing over time. Now, this raises an interesting puzzle. If the way V1 encodes visual stimuli changes continuously, how does the visual system maintain a stable perception of the world? So one suggestion I'd like to describe is that stable perception is maintained through relative similarities of population responses, which, it turns out, remain stable. And the way we tested this is, again, by simulating responses to a bunch of images according to the model fits for a single session. And we then computed a dissimilarity matrix for that session, which tells us for each pair of images how different the population responses are. So this matrix as a whole gives us some indication as to how V1 represents many stimuli. So, and we repeat that for every session. We compute the dissimilarity matrix for each session, and we then compute the correlation between these dissimilarity matrices. So here, each element in this correlation matrix is the correlation between dissimilarity matrices of two sessions. Okay, and now to test for drift, we look at the correlation between dissimilarity matrices as a function of the number of intervening sessions. Okay, and it turns out that these dissimilarity matrices are stable. Okay, and what this means is that if you think of representations of multiple stimuli as points in multidimensional space, where the dimensions are response amplitudes of different voxels, then the relative location of each stimulus remains stable in that space. Okay, the representation remains close to some stimuli and distant from other stimuli, meaning that the representations are moving in this space, but they're moving together. Okay, the relative similarities remain stable, and that may be the basis for stable perception. Another way to think of this is as a constellation. Okay, say the Big Dipper, right? Every star in the constellation is gradually moving across the heavens, right? But all the stars are moving together, right? Yeah, I don't know if you see the animations. I'm not sure they're working. Okay. Right, so all the stars are moving together, and that conserves the structure of the constellation. It makes it stable. And in an analogous way, although neural representations of individual images are changing, or the responses to those individual images are changing, they're all changing together, preserving the relative similarities between responses to different images. So images that evoke similar neural responses will continue doing so over time, and likewise, images that evoke very different responses. They'll continue to do so. The relative similarities are preserved. And this suggests that maybe the aspects of neural responses that change and drift are not part of the brain's core code for perception. And that instead, the brain relies on relational stability, which enables it to preserve the percept despite changes, ongoing changes in the corresponding representation. So to sum up what we've seen so far, already at the first cortical station of visual processing, representations are changing. And more specifically, some aspects of the representation are changing while others remain stable. Okay, we saw that, yeah, okay. Right, we saw that the response baseline is changing and population responses are changing over time. But other aspects of tuning, such as gain, tuning width, and the stimulus preference, seem to be stable. And finally, the relative similarity between responses to different stimuli remains stable. Okay, so these findings raise many questions, and they also have clear implications for brain-machine interfaces, and more generally for deciphering the neural code. Right, and it's possible, as I said, that representational aspects that are stable are those that are critical for behavior, and those that drift are not. Okay, so for example, we said the neural code might rely critically on relative similarities, not on individual response amplitudes. Okay, and so that was the published part of the talk, and now let's move on to the unpublished part. So yeah, a major issue that we need to address at this point is whether representational drift reflects repetition suppression or adaptation just on longer timescales than previously measured, or whether it is a completely different phenomenon. Okay, so as a reminder, repetition suppression means that when an image is repeated, when it's presented a second time, the brain response will be attenuated. It'll have a lower amplitude than the first time. So here in black, you see the response to different images, and in red is the response to the same image repeated several times. You can see that repeating an image attenuates the response. The response amplitude is smaller. And representational drift studies have repetitions. They have many, many repetitions. So this is what a typical study looks like. You have a single stimulus, okay, for example, a 30-second movie clip, and it's repeated dozens of times within a recording session. And you do the same thing for every session and every day. So clearly, repetition suppression should be considered in those studies. In our study, it's less obvious. Okay, we're using data from a very different design. Each stimulus, as I mentioned, is repeated only 3 times throughout the entire experiment. So there are less repetitions, much less, but still there are repetitions, and that means that there are likely to be repetition effects in the data. Now, because of the way the experiment is designed, later sessions will have more repetitions. So here, I'm showing you in blue the distribution of trials presenting images for the first time. You can see, right, these are new images. You can see that as the sessions progress, there are fewer new images. There are fewer remaining images that haven't been seen earlier in the experiment. And here in orange is the distribution of images shown for a second time. And here in yellow is the distribution of images shown for a third time, which naturally increases as the experiment progresses. So the number of repetitions increases with session number. So maybe those second and third presentation trials exhibit repetition suppression, and maybe that is causing the representational drift. So we need to determine whether repetition suppression can account for representational drift. And we're going to address this in the NSD data that we used in our study, but this question should be asked about every representational drift study. So to test whether repetition suppression can account for drift, we simulated responses, we added in repetition suppression, and then we tested whether that results in effects that look like representational drift. So going back to our model, we take the model fit for a voxel, and we use that fit to predict or to simulate responses to all images in all sessions. So now we're simulating all sessions using the same model, so we know that there's no drift. Right? But now we also take into account whether each trial is the first presentation of an image or a repetition. And if it's a repetition, we scale down the response amplitude by a constant delta. And then we add some Gaussian noise to the responses, and then we take this simulated data and we rerun our analysis. We fit the model to it, and we quantify the model generalizability to test whether we see drift. So first, when delta is 0, there is no repetition suppression. In this case, we don't see drift. Okay, the left panel is using CVR squared to measure goodness of fit. The right panel is using correlation. And there's no drift here. So this was sort of a sanity check because we know that there's no drift in these simulated responses. And in this case, there's also no repetition suppression. Now, when delta is 0.1, we have added repetition suppression into the data. A repeated stimulus evokes only 90% of the original response amplitude. And as a result, we see that model goodness of fit decreases with delta session. So we see drift, but only for CVR squared, not when we use correlation, which is consistent with our empirical results that I presented earlier. And I'll just mention that this is also the case if we add repetition enhancement, okay? That repeated images result in a larger response amplitude. Now, if we normalize each session's mean response, drift disappears, but not if we normalize the variance. Again, it's exactly as we found when we analyzed the empirical data. So this means that repetition suppression can produce effects that seem like representational drift. Okay, so we've made some progress. What we need to ask now is whether repetition suppression actually accounts for representational drift. And our approach will be to analyze data that doesn't include repetitions at all, so that repetition suppression doesn't even come into the picture. So we're going to reanalyze the NSD data using only trials on which images were presented for the first time. Now, the problem is that the number of first presentation trials decreases with session number. And if we were to analyze all of these trials, the number of trials used to train the model would decrease across sessions. And the model goodness of fit would be expected to decrease as well, which again might look like drift. So to keep the number of trials constant across sessions, we subsample each session. We keep an equal number of trials from each session. So in this case, 127 trials. There we go. Okay, so it'll be, in this case, we have 127 trials from each session. And analyzing these trials, we find that there still is drift. Okay, it's a bit hard to see, but there still is drift. It's significant, but only when goodness of fit is measured with CVR squared, but not when measured with correlation. And again, when we normalize the mean response amplitude, the drift goes away. Yet when we normalize the variance, it does not, the drift remains. So using only the first presentation trials results in very similar effects as our original analysis that used all trials. So this means that the representational drift that we found is not the result of repetition effects. We can also, we can change this a bit. We can include only the first 20 sessions instead of the first 30 sessions. And this way, we can use more trials from each included session. And in this case, we see similar results. There's drift when measured with CVR squared, but not with Pearson correlation. We can even limit ourselves to the first 12 sessions, in which case, again, we have more trials per session, 358 trials, and we still see the same pattern. Okay, there still is drift. So when we analyze only the first presentation trials, we get the pattern of results that we got when analyzing the whole data set. And this means that the drift is not a result of repetition suppression. We can run the same analysis on the second presentations, ignoring all the first presentations and third presentations, and we subsample all sessions to have an equal number of trials, 136 trials per session. And we again find significant representational drift when measured with CVR squared, but not with correlation. So this is basically a replication of the analysis of the first presentation, but note that we're using a different partition of the data. Okay, it's entirely different trials. And again, we can be sure that this effect is not the result of repetition suppression. And finally, we can try running the same analysis, just to complete the picture, using only the third presentations. In this case, we use only 31 trials from each session, and we still see drift. Now, in any case, we find drift in the NSD data even in our trials. And I also showed you that we reach very similar results using different disjoint or non-overlapping partitions of the data set, which also shows that this is quite a robust effect. So that was the first approach, okay, to analyze particular partitions of the NSD data set. Our second approach is to leave the NSD data and analyze an entirely different data set. The things data set is a multimodal data set that includes fMRI, MEG, and behavioral data sets. So we analyzed the fMRI data set using the exact same approach we used for the NSD data set. And what's really great about this data set for our study is that it doesn't have any repetitions. Okay, or more accurately, most of the images have no repetitions, and those are the ones we're analyzing. So we ran the same analysis, fitting the model to each voxel in V1 and cross-validating cross sessions. And in two out of three subjects, we find significant drift. The average over subjects is not significant, which isn't particularly surprising because this has much less data than the NSD. Okay, only 12 sessions and only 3 subjects. But we see the drift in the 2 subjects only when measuring goodness of fit with CVR squared and not with correlation. Again, this indicates that the changes are such that correlation ignores them, such as an additive change, a baseline shift. And indeed, when we normalize the mean, the drift disappears. Okay, so this is sort of a replication of our main findings of representational drift in a different data set, and it also shows that the drift cannot be accounted for by repetition suppression, since in this case there are no repetitions. Okay, so to wrap it up, we saw that representations in human visual cortex change over time. We saw this in the natural scenes data set and also in separate disjoint partitions of that data set. And we saw evidence of drift to a certain extent also in the things data set. And we saw that the drift is the result of changes in baseline responses. We also saw that repetition suppression should be considered seriously in studies of drift, even when using our approach of model cross-validation. Nevertheless, we were able to identify representational drift that cannot be dependent on repetitions, since we saw it when using data with no repetitions. Okay, but I think it's still unclear whether other reports of representational drift are independent of repetition suppression. Okay, but we can conclude from this that there are multiple parallel processes that change neural representations at varying time scales. Okay, multiple processes such as representational drift and repetition suppression. And these different processes are likely to have entirely different functions. Okay, I want to end. I want to end with the words of Heraclitus: "Everything changes, and nothing stands still." Right, and you can't cross the same river twice. I want to point out that the fact that representations change might not be a big surprise. in fact, when we take into account, for example, synaptic turnover and just general neural plasticity. And I know that some people expect neural responses or representations to change over time. And there are also computational considerations that might support drifting representations. But what's really exciting for me is that we're actually able to measure these changes non-invasively in the human brain. And not only are we able to capture these changes, we're also able to pinpoint what exactly is changing in the tuning functions of voxels or neurons in visual cortex. So for me, that's pretty amazing. And I just want to mention that we are looking for new lab members for PhD or postdoc positions. So if this sounds interesting, give me a call or drop me a line. And I want to thank my postdoc advisor, Eli Miriam, with whom I did this work. I want to thank you, Shawn, for inviting me, and thank you all for listening.
Zvi, thank you very much. This was a really exciting talk, and I want to invite everybody to unmute yourself and to give Zvi a big a big applaud. And I also want to open. Wait, Zvi, is it okay for you to answer questions?
Absolutely, yeah. That's
Before
why I'm here.
I Okay, so so I'd be happy to open the floor for questions, and and of course I have questions myself, but okay, so I'll start. I wanted to ask, so first of all, it's really weird. I mean, the fact that the baseline changes, especially in V1, and I wonder if you think that similar effects are taking place in say in V2 and V3 and maybe high order areas, or I mean, because it does sound like for high order areas, they would be catching up on the what you said, the relational topology or the relations between them. That does make sense, but retinotopic areas, I'm wondering how they would I mean, are you looking into? I mean, it sounds like there's infinite questions that are stemming from your findings, and so that's my first question. Yeah.
Okay. Yeah, absolutely. An infinite questions stemming from this. I agree. Yeah, and you raised several points. So about first of all, just V2, V3, V4, we're working on that. We're both using this steerable pyramid model for the other regions, testing whether there's drift there. And we are also, right, the problem with that is that this model is appropriate for V1, but less so for as we move down the hierarchy. So we're also using well, for example, the texture model, Simoncelli's texture model for V2, which can also be used maybe for V4. So we're working on that as well. And for higher order areas, we're still not sure to use for that. But we are definitely testing models on V2, V3, and V4. And you raised the question of retinotopic areas. Let's just say that if it's just a baseline change, that shouldn't change the retinotopy of a voxel, right? So the
Yeah.
preferred location should stay the same. We're talking about other changes that won't change the retinotopic map.
So at the moment I see that, can I go on? Yeah, that's really interesting. So it's fascinating. I mean, I didn't think of the fact that you would need different types of models for different areas, which makes a lot of sense. So I wanted to ask if the effect sizes, I mean, you checked for the no repetition, the one repetition and the two repetitions. Sorry, for the first time, the second time, and the third time. And I wonder if you see a difference in the size of the effect. I mean, do you think that there is some contribution of repetition suppression at all, or is this changing as a function of the repetition of the image, or you think it's completely independent of whether the image was repeated or not?
Okay, so you're asking first of all whether we do see repetition suppression, and second, basically if they interact.
You showed that there isn't, but I'm wondering
No,
whether
okay, no.
yeah.
So yeah, we do have some evidence for effects of repetition suppression in the data. I'm just showing that this when we remove those effects, we still see representational drift.
Mm.
I don't, Yeah, we still have a few questions we need to ask there. First of all, whether, probably part of what we measured originally, or no, probably maybe some of what we measured originally and attributed to drift might actually be repetition suppression. I don't know if there's an interaction. It's an interesting question.
Yeah.
Um, and about the effect size for different presentations, nothing sort of jumped out, but keep in mind that at least what I've shown here was using different number of trials for each presentation. Like
Yeah.
for the third presentations, we're really limited. And so the SNR there is very low. But we could, that would be an interesting idea. We could equalize the number of trials for each presentation and compare them. We haven't done that. Yeah.
Yeah. I mean, I don't know if that's going to lead to, but yeah, I was thinking whether that because intuitively or when you're looking at the very basic time scales that people usually look into, not across sessions, you don't think that V1 would undergo repetition suppression, like a blink would be a refresh or restart for the activation. And this is kind of like surprising that the baseline changes. And I was trying to think whether maybe it has to do with some like redundancy in the representation so that you have excessive just ideas. Maybe you have excessive representation of the visual image, not only by loads of photoreceptors, but also in V1. And maybe maybe there's some ups and downs of who's now contributing to the representation at the moment. So maybe it's not all the same neurons, but maybe some are. Do you understand what I'm saying? Like maybe there's some on and off states of neurons, and maybe some are at the moment, they're kind of like under fatigue phase, and in another session or another day, they are in the active phase. So maybe, the same orientation and, the location in space is represented by not multiple columns, but multiple neurons that have redundancy, and then this on and off interplay changes the baseline in some sense because you do have some aggregation of activity to that results in the fMRI data. I'm just
Yeah, I think that's really interesting. Yeah, there could be some sort of role switching between different populations that are active at some times. They all represent images, other times other populations. And I'll just point out, just to be clear, that while some voxels, I mean, it's a baseline change, so some voxels increase their baseline, others decrease, and so yeah, that would be consistent with an idea, the idea that you raised, I think.
yeah, really interesting. Addition, yeah, I mean, I've asked quite a lot. So um, okay. So I see that there are no, I mean, actually, I do have additional question I'd like to ask, but I don't think I'll email you about that. Because I think this method can be applied. I mean, actually, not only to vision, which but to many existing data and data sets. And especially, it would be interesting to examine data that did um, um, employ adaptation effects to see what happens there. I mean, if you could apply it. Or do you think that this is only occurring at a cross sessions? Do you think if you would check drift, would you be able to tease apart um, adaptation effects and representational drift even within a session? Or
Yeah, so in the paper I briefly mentioned at the beginning from Yaniv Ziv's lab,
Mhm.
those changes are within sessions mainly. They're also between sessions, but also within. And they're aware of the potential problem of adaptation, so they have this method where they ignore the first repetitions and wait for sort of for the signal to reach some steady state, and they analyze what's left after that when there seems to be no adaptation at that stage, and they still see
The drift.
representational drift. Yeah, so Um, it does seem that these are two phenomena that can coexist, and they also seem to be, again, as I said, sort of at different time scales, even within a session.
Wow, interesting, really interesting. I want to read the comments in the chat. So Yuval Katz wrote, "Great talk. Thank you, Zvi. I was wondering whether, and if so, how representational drift relates to behavioral responses. Example, does it improve, impair object detection thresholds and sensitivity?" So that's a question that within the chat. If you're, I don't know if you have time, we're happy to answer that.
I'm happy, yes, happy to. And I think we have time also, yeah. Um, yeah, the relation to behavior is interesting and a bit tricky. Basically, we don't know. We don't know how representational drift relates to behavior, at least in visual cortex. The assumption usually is just that behavior is stable and that these changes are, they don't have behavioral implications, but there have been questions raised about that. Maybe behavior hasn't been measured well enough in these studies. But we don't know. We don't know. Basically, we don't know the relationship, whether it improves or impairs, sensitivity. We don't know, and those are very interesting questions. I just want to point out, Roy Shul had a great study on behavior over long time scales. Um, and I think that might be very relevant to this question. Worth looking at that study.
Okay, so I'm not sure Yuval is yeah, but that sounds exciting, and I will we can notify him about this. And Lucia Garrido says, thank you so much. That was really interesting. Um, so I want to thank you, Zvi, for a really interesting and exciting talk, and I'm sure that many people will be interesting to collaborate with you and investigate this phenomena or phenomenon. I don't even know if it's one or many. Um, yeah, effects. Really interesting, and thank you so much for joining us today.
Okay, thanks a
Um,
lot, Shalom.
it was a pleasure. And next week, if everything, so Aya Ben Yaakov is supposed to be giving a talk, but we will send details, yeah, in the next week or so. And I hope that you all have good and safe and peaceful summer. And thank you, Tzvi, again so much.
Thank you.